Imagine if you could, I don't know, freeze time in a bustling city.
Okay.
And you could peel back the roofs of every single building and look directly at the open books sitting on every person's desk.
That would be quite the view.
Right. You wouldn't just know where the buildings are, you'd know exactly what everyone inside is planning to do next. And, well, that level of predictive, hyperspecific mapping is what scientists have actually just achieved.
Yeah, but they aren't looking at cities, obviously.
No. They are looking at the intricate architecture of human and animal tissue, right down to the individual cells.
It really is. I mean, it's a profound leap in how we study biology, because we've been able to see the buildings of a tissue for a while now.
Right.
But reading those open books to understand a cell's intentions, all while preserving the entire neighborhood, that takes us into completely uncharted territory.
Well, welcome to another deep dive. Today, we are exploring this massive breakthrough published in Nature Biotechnology by Llorens-Bobadilla and a really brilliant team of researchers.
Yeah, they've introduced a technology called spatial ATAC.
And we are going to unpack exactly how it works, detail by detail, and explore how it reads the open instructions of our DNA while preserving the actual physical geography of the tissue.
And we really have to emphasize why that geographical landscape matters so much.
Yeah.
In biology, location dictates function.
Yeah.
Where a cell sits in an embryo determines what organ it becomes.
Exactly.
And where an immune cell sits in a tumor determines whether it can actually fight the cancer.
So whether you are a biology buff or you're just, insanely curious about how life develops and how diseases like cancer operate in a physical space stick around, this is basically a masterclass in reading the genome's most hidden layer.
To really appreciate the sheer elegance of what spatial ATAC does, though, we first need to understand the incredibly frustrating biological limitation that it fixes.
Right, the old way of doing things.
Yeah, we need to talk about chromatin and what biologists call the epigenome.
Okay, let's unpack this because I think it's a really common misconception that our DNA is just like a long, loose string of code just floating around inside the nucleus of a cell.
Just waiting to be read.
Yeah, exactly. Readily available at any time.
Far from it. If you took the DNA from just one of your microscopic cells and stretched it out end to end, it would be roughly 2 meters long.
2 meters?
Yeah.
From a single invisible cell.
Right. So to pack 2 meters of genetic code into a nucleus that is a fraction of a millimeter wide, the cell has to wrap that DNA around specialized protein complexes called histones.
Okay, histones.
Think of them like microscopic spools. The DNA is tightly coiled, stacked, and compacted. And that massive DNA protein complex is what we call chromatin.
And the physical reality of that compaction is the key here, right?
Yes, exactly.
Because the cell's machinery, like the enzymes that actually read the genes to build proteins, they physically cannot access tightly packed DNA. It's locked away.
It is quite literally a closed book.
So for a gene to be activated, that specific section of the chromatin has to physically unspool. It has to open up.
That's the core of it. We refer to that state as chromatin accessibility. And this accessible open chromatin is the epigenome.
It's the regulatory instructions.
Yeah, it's the regulatory layer sitting on top of the DNA blueprint, dictating which parts of the manual are actually open for business. Mapping these open regions gives us this ultimate behind-the-scenes look at cell behavior.
Right.
It shows us the invisible control switches.
And for a while now, scientists have used a technique called single-cell ATAC to find these open regions. It uses a modified enzyme to tag only the accessible DNA. But this method comes with a pretty devastating compromise.
A huge one.
To isolate those single cells and read their open DNA, you have to completely dissolve the tissue.
Yeah, it's a destructive process.
Yeah.
You have to use enzymes and mechanical force to just break the tissue down into a liquid suspension of individual cells.
I always think of it like making a fruit smoothie.
Oh, that's a good way to picture it.
Right. Imagine you have this beautifully structured fruit tart. You want to know exactly what fruits are in it, so you throw the whole thing into a blender.
Goodbye, structure.
Exactly. When you drink the smoothie, you can easily identify the molecular profile. You taste the strawberry, you taste the kiwi, the blueberry, but you've completely annihilated the structure of the fruit salad. You have no idea where the strawberry was originally placed in relation to the kiwi.
You lose the physical math entirely. And that analogy perfectly captures the frustration of the last decade in genomics. You get the molecular profile of the cells, but you lose their physical address.
But wait, anyone following the genomic space might be thinking, we already have spatial transcriptomics, right?
Right, mapping RNA in space.
Yeah, mapping RNA, the actual messages being sent out by the DNA while keeping the tissue intact. So why go through the hassle of trying to invent a new tool just to look at the chromatin? Isn't knowing what RNA messages are being made enough?
What's fascinating here is that RNA is just the result. It's the finished product. Open chromatin, on the other hand, represents the potential.
Ah.
Okay. Let's stick with the reading analogy. If RNA is the receipt showing what the cell just purchased, the open chromatin is the open catalog showing everything the cell is currently considering buying.
Oh, wow. That's a great way to look at it.
Mapping RNA tells you what a cell is doing at this exact second, but mapping open chromatin reveals the upstream regulatory networks.
The why and the how.
Exactly. It shows you the master switches and transcription factors that are driving future changes. It shows you a cell's developmental trajectory, before it even starts producing RNA.
So RNA gives you the what, but the epigenome gives you the why.
Precisely.
All right, we are about 5 minutes in, so before we dive into the wild biological engineering of how this new spatial ATAC tool actually works, let's do a quick recap for you listening. We like to do this to make sure the foundation is solid.
Good idea.
So bullet 1, open chromatin equals active DNA instructions. It's the epigenome physically exposing the blueprint.
Yep.
Bullet 2, old methods required blending the tissue into a smoothie to read those instructions, which lost the physical map.
The fruit tart is gone.
And bullet 3, spatial ATAC is the new solution designed to keep the map intact while reading those underlying instructions.
So the immense engineering challenge the team faced was, how do you read the specific open genes inside a cell and simultaneously record exactly where that cell sits in the tissue without moving anything?
The mechanics of how they pulled this off are just fascinating. Let's walk through the protocol because it relies on this really ingenious piece of hardware.
The OMNI glass slides.
Yes. It starts with a tissue sample that is fresh frozen, right? And then it's sliced incredibly thin. We are talking 10 micrometers thin.
Just a translucent sheet of tissue.
Yeah. They take this delicate slice and place it onto that customized glass platform, the OMNI slide. And this slide is doing a lot of heavy lifting.
It's the foundation of the entire spatial map.
Mm-hmm.
Because the surface isn't just plain glass. It is covered in a microscopic grid of tiny, distinct spots.
5,000 spots, to be exact.
Right. And each spot is about 55 micrometers in diameter, spaced 100 micrometers apart. Imagine a massive grid of landing pads. And inside every single one of those microscopic landing pads are millions of synthetic DNA strands.
And these strands aren't just random, right?
No. They are highly structured oligonucleotides. Crucially, every strand within a specific spot contains a unique sequence of genetic code. It's a 16-nucleotide sequence that acts as a spatial barcode.
A molecular GPS tag.
Exactly. Yeah. Every single landing pad on the grid has a different barcode. So the logic is, if you can somehow attach that barcode to the open DNA in the tissue sitting right above it, you'll know exactly which geographical coordinate on the grid that DNA came from.
Okay, so we have the tissue slice sitting directly on top of this grid of GPS tags.
Right.
The next step is locking the tissue down. They treat it with a formaldehyde solution to cross-link it.
Which is a crucial step.
Right, because formaldehyde basically acts as a chemical fixative. It freezes everything in place so nothing drifts away.
It creates rigid bonds between the proteins in the DNA. It structurally solidifies the tissue so that the delicate open chromatin doesn't collapse or move during the chemical reactions they have to do next.
And at this point, they immunostain it and take a high-resolution photograph of the tissue, right?
Yeah, to get a visual baseline map of the cells before they do anything else.
Then we get to the really clever part. They permeabilize the tissue, basically poking microscopic holes in the cell membranes, and they send in the biological scissors.
The custom TN5 transposase.
Yeah. And TN5 is wild. It's this naturally occurring enzyme that they've hijacked.
Transposases usually help DNA move around. But here, the custom TN5 acts as a highly selective pair of molecular scissors. Because the tissue is cross-linked and fixed, the tightly wound DNA remains completely impenetrable.
Right, the closed books.
The TN5 scissors physically cannot access it. They can only latch onto and cut the DNA that is naturally spooled open.
So it selectively snips the open instructions, and as it cuts, it performs a process called tagmentation.
Tagmentation, right?
It leaves a little genetic adapter, or basically a chemical handle attached to the ends of the freshly cut open chromatin.
But notice the spatial problem we still have here.
Oh, right.
This cutting and tagging is happening directly inside the nucleus of the intact cells. The open books are being tagged right where they sit, up top, but the GPS barcodes are sitting on the glass slide underneath the cells.
We have the cut DNA up top, the GPS tags on the bottom, and we need to link them together permanently.
And they don't just naturally fuse. So the researchers created this stroke of chemical genius to bridge the gap. They call it a chimeric splint oligonucleotide.
Chimeric, just meaning it's a hybrid built from two different things.
Exactly. The design of this splint is so elegant. It is a custom piece of DNA where one half perfectly matches the chemical handle that the TN5 scissors just attached to the open chromatin, and the other half perfectly matches the sequence of the GPS barcode sitting on the glass slide.
Oh, wow. So it is quite literally a molecular bridge, like double-sided tape.
Perfect analogy. They flow this splint into the tissue while simultaneously doing a very gentle digestion of the cell structure using a protein-melting enzyme.
So they soften the scaffolding just a little bit?
Right. As the cellular scaffolding slightly relaxes, those cut pieces of open chromatin sort of drop down onto the slide. And the splint acts like a specialized docking mechanism.
It grabs the chromatin fragment coming down from the cell with one hand, and it grabs the GPS barcode on the slide with the other.
Yes. And once they are docked, they add a polymerase enzyme to fill in any missing genetic gaps and a ligase enzyme to permanently seal the whole structure together.
Ligation and gap fill. And just like that, every single piece of open chromatin now has a permanent GPS barcode attached to it.
It's beautiful chemistry.
The tissue can be washed away, the DNA is collected and sequenced, and a computer simply reads the DNA, sees which gene was open, reads the attached barcode, and plots it perfectly back onto the original physical coordinate.
It allows us to map the unmappable. We finally have the regulatory switches and the geography at the exact same time.
It really is breathtaking biology. So we have this incredibly powerful tool. But a tool is only as good as what it actually reveals.
Right. What do you point it at?
Exactly. If we have a technology that maps potential, the ultimate test is applying it to a system where biological potential is turning into reality at breakneck speed. I'm talking about a developing embryo.
And the researchers knew this was the perfect proving ground. They applied spatial ATAC to mouse embryos at 3 critical stages of gestation.
Days E12.5, E13.5, and E15.5, right?
Yes. This is a window known as organogenesis. It's when the undifferentiated blueprint of the animal is aggressively complexifying into distinct specialized organs.
The sheer amount of data they pulled from these tiny cross-sections is staggering. They captured up to 7,100 unique fragments per 55 micrometer spot.
Which is incredible resolution.
But what fascinated me is how the algorithm handled it. They fed all this genetic sequencing data into their computer models. Now, the computer doesn't know what a mouse embryo looks like. It doesn't know what a brain or a liver is.
Right, it's just looking at numbers.
It just looks for patterns in the open chromatin and groups similar GPS spots together into clusters.
And when they projected those mathematically derived clusters back onto the spatial grid, they perfectly recreated the anatomical landmarks of the developing embryo.
It found 11 main anatomical landmarks. The algorithm drew the anatomy blindly, purely by reading the open genetic potential.
Let's talk about the specific biology they found, because it proves why looking at the epigenome is so powerful. In the clusters that physically map to the developing brain, the central nervous system, they found massive accessibility in genes like SOX1 and FOXG1.
And those aren't just random markers, right?
No, those genes are transcription factors. They are the master regulatory switches for neurogenesis. When SOX1 is physically open, that cell is committing to becoming part of the nervous system.
Wow.
The algorithm saw the exact spatial domain where the blueprint for the brain was being heavily referenced.
And we saw the same logic elsewhere, too. In the tissue destined to become bone and muscle, the mesenchyme, the MY9 and SMAD3 genes were wide open.
Right.
And in the cluster mapping to the fetal liver, the H-by-A1 gene was highly accessible.
Which is a really beautiful detail, if you think about it. H-by-A1 is involved in producing hemoglobin.
Wait, hemoglobin? Why would it be open in a developing liver?
Because at that specific stage of embryonic development, the fetal liver is the primary site for making red blood cells.
Oh.
The epigenome perfectly reflected the temporary physiological function of that specific spatial location.
Okay, here's where it gets really interesting, though, because I have to play skeptic for a moment.
Go for it.
An embryo is an incredibly chaotic, messy, crowded environment. Cells are constantly dividing, migrating, changing identities. If you look closely at the developing cerebral cortex at day E15.5, it is a densely packed layer cake of biology.
It is very dense.
You have stem cells and maturing neurons physically touching each other. If this technology is melting the tissue down onto these GPS landing pads, doesn't the signal just blur together?
Yeah.
Can this tech actually tell the difference between a stem cell and a mature cell if they are sitting right next to each other?
That was the exact stress test the team applied, and the results were phenomenal. They zoomed in on the subventricular zone of the developing cortex.
Okay.
This is a region where progenitor cells, the actively dividing stem cells, reside. Now, these stem cells heavily rely on a protein called SOX2 to maintain their stemness.
To keep them from turning into something else.
Exactly. But just a microscopic distance away, as those cells migrate outward, they mature into fully formed neurons, and they shut down that SOX2 pathway.
So they are geographically right next door to each other, but biologically they are in completely different universes.
And spatial ATAC cleanly separated them. The GPS spots covering the progenitor region showed wide-open chromatin around stem cell maintenance genes. Meanwhile, the spots just next door covering the mature neurons showed those stem cell genes tightly closed.
Wow, they shut the book.
Yep. And accessibility shifted entirely to genes responsible for building neural connections. The technology successfully traced the exact spatiotemporal timeline of a cell maturing, all while preserving its exact physical location in the dense brain structure.
So it didn't blur, it caught the exact gradient of cellular development.
It really did.
Amazing. Okay, let's do our second quick recap before we make the leap to human medicine.
Sounds good.
So, bullet 1. The technology uses TN5 biological scissors to snip open DNA and a clever chemical splint to bridge that cut DNA onto physical GPS barcodes.
Ligation and gap fill, exactly.
And bullet 2. This allows us to literally see the biological instruction manuals, like the SOX1 gene for the brain or HBA1 for the liver turning on in real-time space without losing the anatomical picture.
Perfect summary. And that transition from understanding basic developmental biology to tackling human medicine is where this research gets highly translational.
Right.
Because mapping a mouse embryo is a brilliant proof of concept, but the ultimate goal of genomics is understanding and solving human disease.
And that brings us to the clinical case in the paper, the clinical pathology. The team took this exact same spatial ATAC technology and applied it to a human breast cancer sample. They looked at tissue from a patient with treatment-naive invasive ductal carcinoma, specifically HER2 positive breast cancer.
And treatment-naive is a crucial detail there. It means the tumor hasn't been altered by chemotherapy or radiation yet. The researchers were looking at the cancer in its natural, uninhibited state.
Right. And for some quick context for you listening, HER2 is a protein that sits on the surface of some cancer cells. It basically acts like a broken satellite dish that is constantly receiving a signal telling the cell to divide and grow aggressively.
It's a major driver of the cancer.
Yeah. Identifying if a breast cancer is HER2 positive is critical for deciding how to treat the patient. So when they laid this tumor onto the spatial grid, what did the epigenome actually reveal?
Well, initially, they had a human pathologist manually analyze a slice of the tumor under a microscope and draw physical boundaries. The pathologist mapped out the invasive cancer, the surrounding healthy tissue, and the immune cell clusters.
The traditional way.
Exactly. And when the team overlaid the spatial ATAC data onto that same map, the genetic clusters perfectly matched the pathologist's manual annotations.
Which is great validation that the tech works on complex human tissue. But I mean, the whole point of spatial ATAC is that it goes deeper than what the human eye or even a microscope can see.
Much deeper. The technology didn't just map the tumor by looking for the presence of the HER2 protein. It mapped the tumor by finding the exact open, non-coding regulatory regions surrounding the ERB2 gene locus.
Okay, wait, the ERBB2 gene locus, that is the gene inside the nucleus that actually builds those broken HER2 satellite dishes, right?
Exactly. Spatial ATAC allowed us to see the DNA itself propped open, actively driving the engine of cancer. We could see the root cause of the malignancy mapped physically across the tissue.
Finding the tumor's open engine is amazing, but, you know, a tumor doesn't exist in a vacuum. It interacts with the body.
It's an ecosystem.
Yeah. I'm highly interested in the battleground around the tumor. Could the spatial map see the immune system reacting?
It could. And if we connect this to the bigger picture, it provides perhaps the most exciting insight in the entire study.
Okay.
The technology identified a highly specific cluster of immune cells, specifically myeloid cells, by detecting uniquely open chromatin at a gene locus called C1QB.
And myeloid cells are essentially the foot soldiers of the immune system. They are often the first responders to tissue damage or rogue cells, but where exactly were they located on the map?
They weren't scattered randomly throughout the healthy tissue. They were massed right at the immediate interface of the tumor microenvironment.
They were lined up at the border.
Exactly.
That is the power of spatial context. We aren't just seeing that immune cells are present somewhere in a blended smoothie of tissue. We are seeing them physically engaged at the front line.
And because we're reading their open chromatin, we can start to understand what they're actually doing. Are they actively fighting the tumor? Or has the tumor chemically hijacked them to help it grow? Right. The epigenome holds those answers. It represents a totally new lens for spatial functional genomics. For decades, the actual control switches of the DNA have been a black box when it comes to spatial geography.
It was always one or the other.
Yeah. We either had the beautiful microscopic geography without knowing which genetic switches were flipped, or we knew exactly which switches were flipped but had no idea where those cells lived in the tissue. Spatial ATAC bridges that gap.
So what does this all mean? It allows us to understand exactly how local environments, whether it's neighboring cells in a developing brain or the immune foot soldiers bordering a tumor, physically alter the operating instructions of our DNA.
Exactly.
It's the difference between knowing that a light switch exists in a house and actually seeing who is standing in which room, flipping it on and off.
That's a perfect way to put it.
Think about what that means for you and for the future of medicine. I want to leave you with a final thought to chew on today.
Let's hear it.
If we can now read the exact open genetic instructions of a single tumor cell hidden within a mass of healthy tissue, and we know its exact physical zip code, well, could we one day design gene therapies that act like molecular Trojan horses?
That's an interesting thought.
Right. Imagine a therapy that circulates harmlessly through your entire body, completely inert, until it physically encounters and reads that specific, localized epigenetic barcode that is unique to the tumor environment. It only unlocks its payload when it confirms the location and the open DNA of the target.
That is the kind of breathtaking precision this technology is making possible. It's a horizon where medical treatment becomes as complex and as precise as the map of life itself.
Keep questioning the world around you, keep looking for those open books, and thank you so much for joining us on this deep dive.